Welcome to Study Loop.
Hi everyone, my name is Eric, and let's go over problem 16. In problem 16, our focus is going to be on a poly-A sequence. This is also known as a polyadenylation sequence. And what does this mean? A poly-A sequence is essentially a string of A's. A's being adenine, right? And the process of adding adenines is adenylation. This is part of the post modifications made to an mRNA. So after transcription, we make an mRNA, and this mRNA has to be processed, right? So this mRNA is going to be made in the nucleus because transcription occurs in the nucleus. So if we drew a picture here, if this is our nucleus, our mRNA would be made in the nucleus, right? Because that is where the DNA is located. This mRNA would need to be modified so that it can be shipped out into the cytoplasm. So modifications occur inside of the nucleus. So as we can tell from your answer choices, C and D are not relevant because it is not in the cytoplasm where poly-A sequences are added. Okay, so all we're left with is A and B. Now let's figure out which end it's added to. The poly-A sequence is added to the mRNA, and as we know, the mRNA is going to be having two different sides to it. So on the left, we're going to have a 5' end, and on the right, we have a 3' end. Okay? And so the part of the modifications is that there are two different ends that need to be packaged. So the first end is going to be the 5' end. In the 5' end, we add what's called a 5' cap. So this is our cap. Now this cap is going to actually lead us out of the nucleus into the cytoplasm. Additionally, on the other side, we have the poly-A sequence. So it's literally a string of A's that are added to the end of the mRNA sequence. And so, from your answer choices, we have A and B, and the correct answer for this is going to be B, where we are adding it to the 3' end of the transcript. And thus it is located in the nucleus because it needs to be modified to be shipped out into the cytoplasm where translation can occur. And that's where the ribosomes are actually located so that we can make our protein. Okay, so I hope this helps the explanation of the poly-A sequence and the modifications that occur to the mRNA. Thanks for watching and see you next time.